Hey everybody, welcome back to another video. In today's video, we're going to talk about the immune system. This video is going to be a complete introduction on the immune system. If you've been wanting to learn more about the immune system, or if you're in an introductory course on immunology, then it is a great introductory video. We will dig into everything immune system, all of the major functions and the tiny little details. Now, of course, we won't cover every single thing. That will be covered in my advanced immunology playlist. Okay, so if you enjoy this video, consider liking, consider subscribing, and maybe leaving a comment. Okay, without further ado, let's get started. So, what is the immune system and what does it do? Well, the immune system is made up of many moving parts. It's made up of leukocytes, which are white blood cells, and we'll talk about all the different kinds of white blood cells today. It's made up of a whole bunch of, immune organs. Okay, we'll talk about them. And it's also not just the cells and the immunogens, it is also just barriers and stuff. For example, your skin can have an immune function by just preventing things from coming into your body. Okay, so this kind of stuff all comes back to immunity. Now, let's talk about the actual function. So the immune system likes to recognize and destroy, or neutralize, any material within the body that is foreign. So anything that is not mine, the immune system will destroy. Okay, that's basically skinny on it. Now, this means that it'll start to defend you against any invading pathogen, and we'll talk about what pathogens are. It can remove any worn out or damaged cells or tissues. Okay, it will identify and destroy any abnormal or mutated cells, for example, cancer cells. And it does that through kind of like a seek and destroy strategy, which we call kind of immune surveillance. And unfortunately, sometimes the immune system can mount inappropriate responses, for example, by attacking your own cells or by being hyperactive for no reason, and that can lead to allergies or autoimmune diseases. Okay, and throughout the course of this video, you'll notice that I'll keep mentioning a lot of redundant pathways, and this is because the immune system can be quite dangerous if it's not kept in check. And so we like to keep a lot of redundancy and checks and balances to prevent the immune system from just attacking us randomly. Okay, so when we talk about the immune system, we can actually split it into two arms. There's an arm called the innate, and let's make this bigger. So this is the innate immunity. Okay, and then there is another portion of immunity called the adaptive arm, or the adaptive immunity. Now, the adaptive immunity and the innate immunity are very much so different from one another. They both function to defend you, but they do it in kind of different ways, and they are made up of different cells and different processes. Okay, so we're going to kind of look at the cells here, and then we'll look at some more of the processes in the next slide later in the video. Okay, but innate immunity, you have to know, is non-specific. What do I mean by this? I mean that innate immunity, you're born with this. Okay, the cells in the innate immune system and the responses in the innate immune system, they have always been there. And in fact, evolutionarily, this innate immunity is very much so conserved among many species, while the adaptive immunity is really unique towards vertebrates. Okay, now adaptive immunity is very specific, meaning that it can target extremely specifically the pathogen that it is targeting. Whereas the innate immunity, let's just say you're getting a flu or you're getting infected by something, every time you get infected by that exact same thing, it's almost as if it's a completely new infection to the innate immune system. But the adaptive immune system, not really. The adaptive immune system will recognize that pathogen that it has encountered before because it is very adaptive, okay? And it has memory, and we'll talk about how it actually has that. The other thing is that the adaptive immunity is not really there while you're born. What do I mean by this? I mean that it's kind of acquired, okay? Because the adaptive immunity has a memory, right? When you're first born, you're not really like infected by many things, right? So, because of this, you don't really have that good of an adaptive immunity. However, as you go through life and you keep getting infected by stuff, your adaptive immune system learns and it adapts and it acquires these kind of new memories. It's like, okay, I know that this pathogen is infectious and I know that I can fight it in this way now. And I have memory cells that can remember this and fight it, okay? Again, I'm giving you an overview here, but we'll talk about it in much more detail and it'll make much more sense by the end of the video. So let's dig into, firstly, the innate immune system. What are its components, okay? Well, remember how in the introduction to the blood video, I mentioned that we have white blood cells inside of the buffy coat? Well, those white blood cells that are, again, inside of our circulation in the buffy coat are these guys right here. So let's take a look at what these guys are. So remember, these are inside of the circulation, and they include these cells right here. The first one is called a neutrophil, and don't you worry, we'll look at their functions completely later. The next one is called a basophil. This one is called an eosinophil. Okay, this one is called a monocyte. And this one is called a NK cell, or a natural killer cell. Okay? Now, why called a neutrophil? Well, these neutrophils, they have these little like granules inside of them, okay? And when they discovered this, they found out that these granules are neither acidic or basic. They are just neutral, so they called it a neutrophil. Okay? Eosinophils were named because of the way they were discovered. So when these guys were discovered, they used a red dye that was called eosin, okay? And so they called it eosinophil. Okay. Now, these are the innate immune cells that are present inside of the circulation, okay? Now, there are other innate immune cells, but they're not in the circulation. They are residing in tissues, okay? And you know what we call it when there are cells that are inside of a tissue? We call them resident tissue immune cells, meaning they're just a resident inside of that tissue, okay? Now, there's this one called a dendritic cell. There's this one that's called a mast cell, and this guy that's called a macrophage, okay? Fun fact, the name actually tells you its function. Macro meaning large, phage meaning eater. Okay, so this guy's a large eater, and we'll talk about why and what that even means later. Okay? Alright. One thing I would like you to remember is that inside of the blood, you have to remember that neutrophils are the highest concentration of white blood cells. Okay? Now, if you remember, red blood cells, there's approximately 5 billion of them per 1 milliliter of your blood, okay? With white blood cells inside of the blood, inside the circulation, there's differential count because it really does depend on your kind of immune situation right now, whether you're in an infection or not, okay? Also, red blood cells are very tiny compared to these white blood cells. These guys are extremely huge in size, okay? And like I said in my previous video, white blood cells, they're colorless, okay? Because they don't have, for example, hemoglobin that gives the red color to erythrocytes or red blood cells, okay? So, we have to actually stain them with something to visualize them, okay? And unlike red blood cells, they actually vary in structure. Some of them are big, some of them are small. So yeah, they're very in size, but most of them are actually way bigger than red blood cells. Now, these are the major circulating red blood cells. Now, there are other ones, but for the purpose of this video, yeah, this is fine. Now, you see how these guys over here, they have a weird looking like nucleus, okay? These ones that have a weird looking nucleus are called polymorphonuclear. And because they have these little tiny dots in them, these granules, they're called granulocytes. Okay? Polymorphonuclear granulocytes. Poly meaning multiple, morpho refers to shape, and nuclear is just a nucleus. So multi-shaped nuclei. Look at these guys, multi-shaped nuclei. Polymorphonuclear granulocytes include your neutrophils, your basophils, and your eosinophils. Okay? The ones that are not polymorphonuclear are called mononuclear agranulocyte. Anytime you add an A in front of something, it just means lack of. So they lack granules. Now, as it turns out, this is actually not correct. While this is the name to this day, later on, after naming this, they found out these guys actually do have granules, but they never changed the name, okay? But mononuclear just means that they have one nucleus, okay? And an example of these would be the NK cell, the monocyte, the macrophage, the mast cell, okay? These guys are mononuclear agranulocytes, okay? Now, remember how I said all cells of the blood, they all originate, and in fact, all white blood cells, they all originate from they have one cell inside of the bone marrow. I mentioned this in the introduction to the blood video. Red blood cells come from this one stem cell inside of the bone marrow called the hematopoietic stem cell, okay, or the pluripotent stem cell. That guy also gives rise to every single one of these here, okay? Of course, there are different differentiation pathways, but the main guy at the top is that hematopoietic stem cell. Now, very quickly, the hematopoietic stem cell, that pluripotent stem cell, we know that it can go down something called the myeloid lineage, okay, where this myeloid cell is formed. But also, it can go down a different route, where a lymphoid cell is born, okay? What does a lymphoid cell give rise to? Well, the myeloid cell gives rise to almost everything here. Almost everything here. The lymphoid cell gives rise to almost everything here. Okay? So majority of the white blood cells and your red blood cell are coming from the myeloid lineage, are specifically in the innate immunity. The lymphoid cells are coming from the adaptive immunity. Now, very quickly, there are two types of lymphoid. There is a large lymphocyte, okay, and there is a small lymphocyte, okay. The large lymphocyte is actually referring to the NK cell. The NK cell is a large granular lymphocyte, okay? And it's originating from a lymphoid cell. That's why I have an arrow right here towards it, okay? Now, the other thing is that those small lymphocytes are going to be your, and let me zoom in here, they're going to be your B cell and your T cell. Also sometimes called B lymphocytes or T lymphocytes. Exactly the same thing. So that hematopoietic stem cell inside of the bone marrow, right, will give rise to this lymphoid progenitor, okay, or this lymphoid lineage, and the myeloid lineage. The myeloid would lead to the production of red blood cells, macrophages, monocytes, eosinophils, basophils, neutrophils, dendritic cells, mast cells, while the lymphoid lineage will give us lymphocytes, such as the NK cell, which is part of the innate immunity, and the small lymphocytes like the B cells and T cells, which are part of the adaptive immunity. Okay? Perfect. That's important to remember. Now, we'll touch on this a little bit later, but as we learned, not all of those things stay in the bone marrow. Okay? Obviously, the white blood cells will leave to go into the circulation. Some of them will leave to go into their resident tissues. Okay? And other ones will be completely formed inside of the bone marrow, such as the B cell. Okay? And this B is for bone marrow. They're born inside of the bone marrow and they mature there, and then they leave and they go into the circulation. And then there's other ones that are born inside of the bone marrow, but they actually mature elsewhere. They mature in organs called the thymus, and that's exactly what happens to this T cell. T for thymus. Okay? We'll talk about this a little bit more later. Now, quickly, why do I have an arrow going from a monocyte in the circulation into a macrophage inside of a resident tissue? Well, it's because a monocyte is kind of like a precursor to this macrophage, okay? Where there are resident macrophages that just are chilling or sitting inside of tissues already. However, there are also these monocytes that are just inside of the circulation, and they're precursors of macrophages. Whenever these monocytes come outside of the circulation and they come inside of a tissue, they start to change into a macrophage, and they become better at their function, and they become a little bit bigger as well. They enlarge and they become better at their function, which we'll talk about in a second. Okay? All right. Now, what else can I say? Ooh, this dendritic cell. This dendritic cell is a really important cell, as we'll talk about later. It basically connects the innate immunity and the adaptive immunity. Okay? And this dendritic cell, it has a really special function called antigen presentation. Therefore, they call it antigen presenting cell. If you look at it, it has this protein on its surface called the MHC, which we'll talk about. And this protein is basically expressing on its surface a foreign antigen. Okay? We'll talk about how this guy got the foreign antigen. Okay? That's why they call it antigen presenting cell, because it is literally presenting an antigen on its surface on this protein called the MHC. Okay. And we'll talk about why it's doing this. Now, let's move on and finish with the adaptive immunity, those cells. These B cells, okay, once they become activated, right, when there's an infection and they become activated, they'll start to divide and specialize. They will specialize into this cell called a plasma cell, which looks like this. And this plasma cell can make antibodies. Okay? Look at it, they'll secrete antibodies. But beforehand, these antibodies are actually kind of attached to the cell membrane of this B cell. Okay, before this B cell becomes activated and turns into a plasma cell, those antibodies are kind of attached to the surface of this B cell. And while they are here, they're actually acting like an antigen receptor, meaning that they can bind to antigens. Over here, they're called B cell receptors. Okay? There is another cell that this guy can turn into, this B cell can turn into once it's activated, and that is called a memory cell. Okay? A memory cell. Now, say for example, memory B cell for example, say for example that you got infected with COVID-19 for the first time. Okay? Or you know what, better yet, forget the COVID-19 example. Let's take the flu. You guys know how there's a seasonal flu every year? Well, let's say at the start of the year, you get infected with the flu, right? And you do not get infected with that flu for the rest of the year. Why is it that you don't? It's because of memory cells, okay? The first time that you get infected by that flu, right? These B cells, they become activated. They will bind the antigens, which, by the way, we'll talk about. Antigens are just foreign molecules, okay, that elicit an immune response. Either way, these B cells will bind the antigens of the foreign flu, okay, and they will become activated and turn into these plasma cells and make antibodies. The antibodies will then go and fight off the infection. But while this guy was being made, this B cell made a copy of itself called a memory cell. Basically, this memory cell has these membrane-bound B cell receptors that are really, really specific to that flu virus, okay? So that if that flu virus were to come inside of the body a second time, this memory cell would be there to recognize it, okay? And immediately get rid of it faster than we had to do it the first time around. All right. So that's the concept of a memory B cell, and that's why you don't get sick with the same pathogen twice, okay? Now, some of you might be asking, well, how is it that I get flu every year then? It's because that virus that gives you the flu is mutating itself. And if it does mutate itself and change its shape a little bit, then our adaptive immune system has to adapt and recognize it again, okay? Because remember, these guys are extremely specific. Once they target something, they are really specific to that target. And if that target changes even a tiny bit, these guys are going to have to adapt and change their response and become specific to that thing again. So that is B cells. They make antibodies. And in fact, we can split the adaptive immunity into kind of two parts. We can say that there is a humoral portion, which is mediated by antibodies. Okay? Humoral just means that something is being secreted. And in this case, the thing that's being secreted is the antibodies. The other portion that we can split this adaptive immunity into is called cell-mediated. Now, this will make sense in a second, but this involves these T cells. T cells, they also have something called a T cell receptor. But unlike these B cell receptors, which can readily go ahead and bind an antigen, okay, these T cell receptors, they cannot readily bind the antigen. So you can think of them as blind. They can't actually see the antigen, even though it's right here. It's floating in front of them, but they can't see it. They need somebody to show it to them, okay? And that somebody will be the antigen-presenting cell. The antigen-presenting cell, on its surface protein called the MHC, carries the foreign antigen. And it will go ahead and present it or show it to this T cell receptor. That way, the T cell will become activated and it'll start to divide and differentiate into different kinds of T cells. Okay? Alrighty. We'll talk about this a little bit more later. But in the current stage that they are, these T cells and these B cells, right? In this stage when they're not activated, they are usually called naive. So naive B cell and a naive T cell. Either way, let's say this T cell becomes activated, okay? Who does he turn into? He can turn into a couple of different guys. He can turn into someone called a cytotoxic T cell. This one's also called a killer T cell. I think it's pretty accurately named because its job is to literally go stand face to face with the pathogen and just kill it right there. That's why they call it cell-mediated, because the cell itself will mediate the killing of the pathogen, okay? Next is a helper T cell. I like to think of the helper T cell as the general. he is going to tell people what to do and what not to do. Okay? Um, Unlike this cytotoxic T cell, which will directly go and kill things, the helper T cell will not do that. He will just go and tell people what to do. He's the general of the military. This guy is the army. This guy is the general. Okay? All right. And then, of course, we also make a memory T cell for the same reasons we gave here. Okay? Beautiful. Now, sometimes you might see cytotoxic T cells be called CD8 positive, and helper T cells be called CD4 positive. Now, CD4 and CD8, they are just proteins that are present on the surface of these cytotoxic T cells. Okay? So, for example, this right here, this right here, this right here, these are all CD8s. And the reason there's a positive number there is because, well, they're there. CD8 positive. And CD4s are also just a surface protein. So this guy's CD4 positive. And these two kind of help us differentiate between the T cells, right? So if I'm looking at a helper T cell and it has CD4 positive on it, okay, I know it's a helper T cell and not a cytotoxic T cell, okay? And vice versa. They also have other functions, but we won't get into it for this video. Okay, so this is kind of just a basic intro into the adaptive immune cells and the innate immune cells, but you can already kind of see that the adaptive system is just so much more specific, okay? And we'll talk about that a bit more. Alrighty, now before we dive into the functions of all of these guys and what they do, let's quickly talk about the types of pathogens that we just have to kill and encounter. Okay, so first of all, we know that it's not just all about pathogens, right? At the start of the video, I mentioned that we can have cancer cells, right? These are our own cells that have been mutated and are now kind of foreign to us, so we can fight them off. But what about pathogens? Well, we can have things like viruses, okay? We can have fungi or fungus. We can have bacteria. Okay, and we can have parasites. Okay, and I'll give you an example of all of them. Viruses, they can either be RNA viruses or DNA viruses. Now, let me tell you something, okay? Viruses, they are not like bacteria or like us. They cannot carry out metabolism. They cannot reproduce without invading a host cell. They cannot. That's why a virus has to inject its either RNA or its DNA into a cell. And I'll show an example of it in a second. Okay? In order to replicate. Without being able to do this, it cannot replicate or do anything. They are called non-self-sustaining entities. A great example of an RNA virus is the SARS-CoV-2 or the COVID-19. Okay? And a great example of a DNA virus is the HPV or human papillomavirus. Okay? Fungi, you can find fungi in kind of two different categories. One is the Aspergillus, okay? And the other one is Candida, okay? And again, this kind of comes from like yeast. Aspergillus, you can kind of find it growing on multiple different things. You might find Aspergillus flavus, for example, growing on corn, okay? But, you know, it doesn't really matter too much for the purpose of this video. We're just kind of introducing the type of pathogens we have to deal with. Now, it is important to say that as long as you're not immunocompromised, meaning you don't have a messed up immune system, or you're elderly, you can pretty much fight off fungi very easily. Okay? The other thing is bacteria. And bacteria are really interesting. They are single cell, non-nucleated, meaning they don't have a nucleus like us. All right? And they can either be intracellular or extracellular, meaning that extracellular means that they reside outside of the cell and they do their damage to us outside of the cell, like our cells, or they can be intracellular, meaning that they go into our cells and they damage us from within, okay, by being inside of our cells. All right, and a great example of intracellular is it's chlamydia, okay, and the extracellular is E. coli, although certain strains of E. coli, okay. The other thing with bacteria is that they primarily cause damage by releasing either enzymes or toxins that are just kind of going to damage our cells, okay? And the last one is parasites. Parasites, you can have kind of two types here. You can have protozoan parasites, or you can have helminth parasites, helminthic. Helminths are kind of like worms, all right? So this image here is a helminth. Protozoan you can think of as the plasmodium, which is what causes malaria. Okay. Okay, so these are the types of pathogens that we have to fight off, and our immune system has to fight off. By that I mean, okay? Now, whenever we start talking about pathogens, we have to talk about the notion of virulence. Okay, what the hell is virulence? What does it mean? Virulence refers to the inherent ability of a pathogen to cause disease. Okay? The inherent ability of a pathogen to cause disease. It's basically a qualitative description. So, for example, the common cold. Okay? I can say the common cold is less virulent than the parasite that causes malaria. And that would be absolutely true, okay? Because the parasite that causes malaria is has a higher inherent ability to cause disease than the common cold, okay? And again, sometimes you can have the term virulence or avirulence, okay? Something that is not virulent, all right? But virulence is defined as the inherent ability of a pathogen to cause disease. Certain pathogens are just more virulent than others, okay? Great. Alrighty. Alright, now that we know this, let's kind of talk about the functions of all of these immune cells, okay? Well, most of these immune cells. Okay, so let's start with the neutrophil. The neutrophil mainly is able to target bacteria and fungi. If you have watched my previous video on the membrane, you'll know that we talked about the process of endocytosis, specifically a subset called phagocytosis, which means eating, okay? So basically the cell membrane kind of comes out here and forms these arms called pseudopods and then starts eating the material. Neutrophils, monocytes, and macrophages, let me show you. So neutrophils, monocytes, and macrophages are one of the only cells that can perform phagocytosis, okay? Now, macrophages, by the way, do it the best. Now, neutrophils, they are able to phagocytose these fungi, and when you phagocytose, what do? You combine that phagosome with a lysosome inside of the actual cell, and the lysosome will, of course, start breaking down its constituent parts here, and boom, you've killed this guy. Now, neutrophils, they also have another function. They release something called a net, which stands for the neutrophil extracellular trap. Why do I have a picture of a DNA kind of covering this bacteria? Well, it's because they actually quite literally toss out their DNA in the form of chromatin, and they cover all this bacteria here. Why do they do this? Well, because DNA is actually pretty sticky, and this actually traps this guy in its place. Now, this is not the only thing that is released with these nets. There are certain chemicals, such as enzymes or harmful substances that can help digest/destroy this guy, okay? But that's what the neutrophils do. They release nets to kill intracellular bacteria, and they can phagocytose these fungi, okay? All right. Now, next one is basophils. Basophils, man, they are the least understood and the least numerous, by the way, of the white blood cells in our circulation. We know that they kind of have some sort of role in allergy because they release histamine. It's been said that they mediate the immune response to parasites. And also, they release molecules such as heparin, which has been known to be a blood thinner and can speed up fat removal after a fatty meal. But again, we don't know too much about basophils, so don't worry about it too much. This guy, the eosinophil can target helmintic parasites here, okay? And why do I have this parasite, by the way, being covered by antibodies? Well, we know that our B cells, well, specifically, I should say, our plasma cells, which are a differentiated form of our B cells, they're responsible for secreting antibodies. This eosinophil, it is blind to this parasite, okay? This parasite could be standing right in front of it. This eosinophil won't see it. How can it see it? The only way that it can see it is if this plasma cell secretes antibodies that will go and stick to the surface of this parasite. Then, and only then, will this guy be able to see this parasite and release these granules that it has inside of it and cause destruction to this parasite and then kill it, okay? Because these granules, they contain harmful substances to this parasite, okay? So that's what eosinophils do. They're responsible for, parasite killing. The other thing that these eosinophils are involved in is allergic conditions such as asthma and hay fever. The current hypothesis is that allergies are more common in parts of the world with less parasites. And because of this, the eosinophils will kind of become impatient, if you will, and they'll start to release their granules on less important factors, therefore causing allergic responses or hypersensitivities, okay? Now, this is just the current hypothesis. It's not really proven yet. Okay, the next cell we're going to talk about is this big old guy, macrophage here. Remember this guy, the macrophage? He's resident tissue. This macrophage is so good at eating, which is in its name. Macro meaning large, phage meaning eater. Look at him. Big guy. Look at him. He is eating this bacteria right here. Okay? And then, of course, he's going to digest them inside of himself. Now, this guy can do many other things as well. Macrophages are very diverse. They can not only kill substances, so they don't only have cytotoxic function. Cytotoxic just means killing cells. By phagocytosing, for example, they can also secrete these molecules called chemokines, which we'll talk about later, but essentially they help recruit other immune cells out of the blood to come and help. So look, these chemokines released by this macrophage will go to the bloodstream, recruit a neutrophil. Neutrophil will come out of the blood, and then it'll help this guy to start phagocytosing more bacteria. Okay? After this infection is done, okay, the infection is fought off, there's going to be damaged cells. Okay? Think of a war zone. After a war zone, there's always going to be damaged buildings and streets and all that stuff, right? This is the exact same. Your immune cells are going to war. This is life or death, man, for your actual body. If your immune cell loses, then you're dying. So this is exactly what's happening. And after this battle is won, they have to go and repair the cells that were damaged. How do they repair the cells? Well, they will secrete chemicals that will stimulate cell division. And these damaged cells will go through cell division and replace themselves. Okay? Now, you might be saying, okay, what if cells can't go through cell division? Or what if the damage is just too big to replace? Well, that's where scar tissue comes in. And at that point, cells called fibroblasts will come in and secrete collagen to fill up that kind of damaged area. And that's how scar tissue is formed. Okay? Alrighty. The next one we're going to talk about is NK cells. NK cells are like assassins. of the immune system, okay? They don't care. If they see someone that is not of us, and I'm talking anything like cancer, virus, infected cell, they immediately kill it. They immediately kill it. So let's talk about this specific example. Remember our virus here? And remember how I said they either contain an RNA or a DNA? Well, this one contains an RNA, okay? And look at what it's doing. The RNA was originally stored in here, in this kind of capsule, but now it is being kind of wheedled out and inserted into this cell. Why? Because remember, I said these guys are non-self-sustaining. They need us to replicate, okay? So they put their DNA on us and then use our machineries to replicate. Now, what this NK cell does, right, it realizes that this cell is being infected by a virus. It'll release lytic granules. Lytic just means destructive. So it'll destructive granules and specific cytokines. Again, we'll talk about what these are. But these cytokines include interferons or IFNs. And what this does is this prevents or limits the viral replication, okay? And basically kills this virus-infected cell so it doesn't infect anywhere else, okay? So what NK cells do. T cells, and I should be very specific here, cytotoxic T cells, they have a function, which is very crazy. Cytotoxic T cells, after they become activated by, remember, our antigen presenting cell, our dendritic cell, remember, because their T cell receptor cannot just see the antigens, they require this guy to come here and present it to them. But after they become activated and become a cytotoxic T cell, they can pretty much do anything. They can go and kill cancer cells by releasing cytotoxic chemicals. They can go and kill cells that are infected by an intracellular bacteria. Remember how I said they can be intracellular or extracellular? Well, there you go. They can also go and kill virus-infected cells by releasing those same cytotoxic chemicals. Okay? Now, there's one thing that I haven't shown here, but T helper cells, or helper T cells, they can activate plasma cells. Now, you might be thinking, hey, well, hold on. I thought you said that when B cells get activated, they get turned into plasma cells. That's true, but again, this is that part where I said we have redundancies to prevent inappropriate activation of the immune system. When the B cell receptor sees its antigen, right, that it's specific to, it will become activated. And what happens next is that it actually becomes partially activated, and it waits for a secondary activation signal from a T helper cell. And then the T helper cell will come and tell it, okay, listen, you're good to go. You're good to become a plasma cell, right? And then it can become an antibody-producing plasma cell, okay? So this is one of the functions of the T helper cell. Alrighty, so we've talked about all of the functions of these important immune cells. Now let's talk quickly about lymphoid tissues. Remember how I said we have different organs that are kind of associated with the immune system? Well, one of the big one is the lymphatic system, okay? And the lymphatic system kind of just branches off everywhere, okay? Everywhere. And it samples the kind of fluid that surrounds our cells called the interstitial fluid at all times, and it brings it to these areas called the lymph nodes. These lymph nodes are so important, man. These lymph nodes, inside of them, have macrophages, resident tissue macrophages, that remove pathogens from the lymph. Lymph is just a fluid that flows within the lymphatic system, okay? Just like how blood flows within our blood vessels, lymph flows within lymphatic vessels. They exchange lymph with blood, okay? And there are lymphocytes in here. They call them resident lymphocytes, such as B cells and T cells. The resident lymphocytes make antibodies, and the activated T cells that are in here will go from the lymph to the blood to go and fight off the infection, but only when they're activated. There is another kind of lymphoid tissue called the bone marrow, where, remember, I said that the B cells kind of mature here, and I said that this is the site of the formation of all blood cells, doesn't matter what we're talking about, so they're all formed in the bone marrow, including your red blood cells as well. But some of them mature in different places. For example, the T cells, they mature inside of the thymus, which is a gland that really kind of sits on top of your heart, okay? Alrighty, and this guy actually shrinks with age. Okay, there is other ones called the spleen. The spleen is really good at removing old, worn-out red blood cells by the help of these macrophages. Macrophages that sit inside of this area, they just kind of eat up that old red blood cells, right? Because remember, they live 90 to 120 days. So they have to be removed somehow, and that's done by these macrophages. They just phagocytose them. The spleen also protects against blood-borne pathogens, okay? Because it is filtering the blood. It also exchanges lymphocytes with the blood, okay? And it does store a little bit of red blood cells inside of it, where it can actually be released sometimes into the bloodstream by splenic contractions. There's this other thing called the gut-associated lymphoid tissue, which is really just part of a bigger umbrella term we like to call MALT, or the mucosal associated lymphoid tissue, and that includes the GALT, it includes BALT, so bronchial associated lymphoid tissue, NALT, nasal associated lymphoid tissue, whatever, okay? Many different places have it. But this guy specifically, this GALT, if I zoom in on for example, the small intestine, right? I'll see that here's the cells that make it up, and underneath all these cells, there's this kind of area where immune cells are located, and this area is called the Peyer's patch, okay? It's just an area where immune cells are localized, and they give protection towards the digestive tract, okay? So as you can see, these immune organs, they are strategically placed in areas that are very susceptible towards infections, such as the nasal mucosa, or the surroundings of the cell, which will get picked up by the lymphatic vessels and go to these lymph nodes, okay? Or just the blood, where the spleen is sampling, or the GI tract, okay? There are other ones, such as the adenoid, the tonsils, the appendix. They do really similar things to these lymph nodes, okay? They're all kind of just sampling the fluid environment around the place, looking for something that is not ours. And if it's not ours, then the resident lymphocytes that live within them, these organs, the adenoid, the tonsils, the appendix, or the lymph node, will become activated and will go to work, okay? Now, it is important to remember that in a naive state, these T cells and these B cells are constantly circulating from the blood and then to a lymph tissue, and then to another lymph tissue, and then to a blood, and then to another lymph tissue. They're basically always kind of circulating, surveilling the area, okay, looking for something, okay? And when they get activated, they will kind of meet in lymphoid tissues, such as the lymph node, okay, and interact with one another. Because remember I said the T helper cell has to come and activate this B cell in order for it to become a plasma cell? Well, these two have to find each other, okay? Have to find each other. And imagine, there's one T helper cell in, for example, this lymph node here, and then there's one B cell that has also spotted that antigen in this lymph node. So they have to come and find one another, all right? Okay. So these are all the lymphoid tissues, which again is the site where white blood cells are either stored, produced, or interact with one another, okay? And finally, let's kind of briefly just discuss some of the differences that we have seen between the innate and the adaptive, okay? So The innate response, or the innate immunity, is very fast. It is, in fact, our first line of defense. Okay? So when we get infected, this guy comes in first. It's a very fast response. Within 6 to 12 hours, okay, this guy is kicking in completely. And it is, like I said, it's non-specific. Okay, it doesn't target specific antigens or anything like that. Whereas the adaptive, okay, it's very slow. It's a delayed response, at least the first time, where it takes around 3 to 5 days for it to actually come activated, right? And it is very specific, okay, very, very specific. And we'll dig into that a little bit more in the next slide, and how actually specific it is. Okay, now let's talk about this stuff in greater detail. Oh, actually, one more thing is that, remember how I said all of these cells, they're coming from the bone marrow, right? And some of them mature in different places. Well, majority of these guys can go through cell division on their own and proliferate and blah, blah, blah. But majority of the new lymphocytes, like the T cells and the B cells that you make, are actually through cell division, okay? And oh, actually, you know what? Sorry. These guys, okay, they will be made inside of the red bone marrow, okay? Whereas these guys, while they are made in the red bone marrow and they will mature there and whatever, okay, majority of them are actually made through cell division, all right? And I'll show you what I mean in the next slide, okay? Alrighty. So, here's just the exact same thing we talked about, right? With the innate immunity and the resident tissue cells and all of this stuff, okay? We talked about all of this already. But now I want to introduce you to some more defenses that the innate immunity has, right? Because it has just more than the cells and these resident tissue cells and all of that, okay? It has many different things. One of which is the complement system, okay? The complement system refers to a group of proteins that are made by the liver and secreted into the bloodstream, okay? So these are proteins called complement proteins that are made by liver and secreted into blood, okay? And once they become activated, they will carry out a very specific function, okay? Alrighty, let's see how these guys can actually become activated. There's 3 ways of activating the the complement system, okay? This red protein here, and all these other ones, is a complement protein, okay? And this green guy is a pathogen, such as, a bacteria. Okay, and this brown guy is also a pathogen. Okay, of the 3 ways that these guys can become activated, the fastest way by far is a spontaneous kind of collision, okay, or spontaneous binding of this protein, this complement protein, onto the surface of this bacterial or pathogen or microbe, okay? So spontaneously just binding to it. And as soon as this guy binds, you know what happens? It's kind of like a domino effect. It'll start to activate the other kind of nearby complement proteins around it, okay? So this guy randomly binds spontaneously, and it'll become activated, and then it'll start activating nearby complement proteins. And then those activated complement proteins will activate other nearby complement proteins. It's literally like a giant domino effect, okay? And this is one way of activating it. The system. The second way of activating it, which is, by the way, the slowest, is binding to an antibody. Okay? To antibody. Okay. So remember that our plasma cells will make antibodies, and these antibodies, they like to kind of go and bind to the surface of these pathogens to kind of neutralize them. And again, we'll talk about the functions of an antibody in a second, and you'll understand why they're doing this. Okay, but for now, just realize that they like to do this to neutralize the pathogen so that it doesn't cause us any harm. What these complement proteins can do is that they can come out and actually bind to this region of the antibodies, okay? And when they do this, they become activated, okay? This is the slowest process by which they become activated. The third way is by binding to a carbohydrate on bacterial surface or microbe surface, okay? So you know how our cell membranes, we have glycoproteins or glycolipids, right? And glycose is just like a carbohydrate or sugar residue. Well, it's not just us. Other organisms, microorganisms, they have sugars and carbohydrates on their surface. And what can happen is this guy over here can actually go and bind, this complement protein, it can go and bind to certain sugars that are on the surface of these microorganisms. And when it can do that, it becomes activated, and it can initiate that whole cascade, okay, of activation. Now, if you're curious, the sugar that it binds to is actually called lectin, okay? Mannose. Alrighty. Now, we talked about how they get activated, but what the hell actually happens when the system gets activated? When the system gets activated, two things can occur. One is the formation of something called MAC, okay? So bear with me here. Let's draw a cell membrane, double bilayer, okay? Here's a cell membrane. When these complement proteins become activated, right, no matter in which way, they will start to assemble themselves and insert themselves into the membrane of a microorganism, and they'll form a kind of giant pore, okay? This giant pore that they form is called the MAC, or the membrane attack complex. This giant pore essentially permeabilizes this membrane to everything, meaning things can now go in and outside of this microorganism completely freely. And this is not good because a membrane is there for a reason, and if I just start punching holes all over this membrane, then I'm going to really damage this microorganism, okay? And it can lead to its death, which is called cytolysis, okay? So that's how this complement proteins can kill these bacteria. They can assemble together and form the membrane attack complex. Next is this thing that they can do called opsonization. Okay, what is opsonization? I'm going to have a weird analogy for this one. You guys ever had a piece of toast, just plain toast, okay? It's not bad to eat on its own, right? It's not bad. But imagine you go and grab a jar of Nutella, and you grab that Nutella and you spread it all over that actual toast. Now it's tastier. Now you want to eat it more, and you want to eat it faster. That's exactly what opsonization is. When these complement proteins go and bind to this bacteria, right, is going to increase the rate of phagocytosis. How so? Well, essentially, it makes the bacteria or the microorganisms seem more tasty to our phagocytes, such as neutrophils and macrophages. Okay? So that's what opsonization is. It is tagging the actual microorganism and making it look more tasty for phagocytosis by our macrophage or our neutrophils, okay? And it increases the rate of that. Alrighty, so that's the complement system, okay? And that's how the complement system helps defend us. And again, it's all part of the innate system, okay? The other thing is these interferons, which I mentioned before. Interferons are just a class of molecules that can be released by NK cells or viral infected cells. meaning that a virus has infected this cell and they start releasing interferons. What do? Interferons, all they do is limit viral replication. Easy. That's it. That's all you have to know. They limit viral replication. Okay? The next thing that occurs within innate immunity is inflammation. This process of inflammation, we'll talk about in more detail a little bit later in the video, but for now, just realize that this process of inflammation has 4 characteristic symptoms, which I'm sure all of you have experienced. First one being heat, right? So when you touch the area, it's a little bit hot. Next, there's maybe a little bit of redness to the area. There is some swelling. And finally, there's some pain. Okay? The only thing you have to realize is that while the severity of inflammation can vary, the underlying mechanism or process is the exact same. Okay? It's literally the same response, but the severity can, of course, vary. Okay? And of course, you guys understand that when we talk about inflammation, right, sometimes it gets a bad rep. And we have to differentiate between acute versus chronic inflammation, because chronic inflammation is bad. Okay, chronic inflammation is like giving you diseases such as like obesity or diabetes or type 2 specifically, atherosclerosis, right? All of these diseases are characterized by chronic inflammation. Some others include like rheumatoid arthritis, whatever. Acute inflammation is good, okay? And its entire goal is to just clear the pathogen, kill it, okay, and then initiate the repair response, right? All right. So this right here is all of the innate immune responses that we have to kind of prevent infection or kind of clear that infection, okay? Now, like I said, there are other things, like there's external barriers, like, tight junctions that are forming between your skin cells, tears, cilia, or your nose hairs, right? Mucus linings, acidity, all this kind of stuff, it exists as like a barrier. There's also even antimicrobial defenses where we kind of secrete like proteins and stuff that will just try to damage bacteria before our immune cells even have to do anything. So there's many things that are part of the innate immunity, but this is the main stuff that I would like you to know. Now, let's finish up our discussion on the adaptive immune system, because we left a couple of things unanswered. One was that how do these B cell receptors work and what do they look like and what's their function, right, alongside these antibodies, and these T cell receptors, and what's their deal about not being able to see antigens when they're right in front of them. Okay, so first things first, let's go ahead and take a zoom in on what these guys actually look like. Okay, so here is going to be an example of a B cell receptor slash antibody. Okay, and we're just going to go through some anatomical landmarks very quickly. One is that, as you can see, this right here is the antigen, and this portion right here then must be the antigen binding site. So antigen binding site, which is right here and right here. Okay, they have this region right here called the Fab region, which honestly you don't have to know, but it stands for the fragment antigen binding region. Okay, they have this other region stretching from here to here, which is called FC, or fragment crystalline. Okay, and this portion right here carries many of these secondary functions. For example, when the complement proteins bind, they bind to this FC region. Okay, or remember how I mentioned that the eosinophils need to see this actual parasite? Well, they have to bind to this Fc region, okay, to see it. Okay, alrighty. Next is going to be the fact that this thing is made up of two portions, this guy and this guy. And this dude over here is much lighter than this big guy over here. So you know what we call it? We call it the light chain, based off its weight, and we call this bigger guy here the heavy chain. Okay. Alrighty. Now the antibody has three functions. One is called neutralization. The other one is called opsonization. Okay. And the last one is called complement activation. So we've already seen one of these, which is complement activation, right here. The antibody binds, okay, to the surface of the pathogen, and then complement will come and bind to it and become activated. So that's one of its functions, okay? The second function is opsonization. So just like the complement proteins can opsonize and increase phagocytosis, antibodies can do the exact same thing. They can make the pathogen look more tasty for our phagocytes, okay? The last thing is neutralization. This is a very interesting function where essentially neutralization is talking about the fact that this antibody will be binding and blocking any viral or bacterial proteins that normally try to interact with our cells, or, you know, binding or blocking any toxins that these bacteria release to try and damage our cells. And this is, of course, going to inhibit bacterial or viral access to the cell or damaging of our cell, right? So that's why they call it neutralization. Okay, you neutralize the threat. Okay, so that's that. These are the functions of an antibody. The next thing we're going to look at is this T cell receptor. What does it actually look like? So this T cell receptor, right here, T cell receptor, it has this region over here, which is the antigen binding site. Okay. It has this region over here, which is called the variable region. And this is a very complicated answer as to why it's called the variable region, so I'm not going to explain that, at least for this video. And then constant region here, and then of course a region which is membrane bound, because all receptors have to have a membrane portion, right? Okay. And you might be asking, hey, where's the membrane bound region for this one? Well, this image is of an antibody specifically, but if it was a B cell receptor as well, it could also have that membrane bound region, okay? And by the way, the morphology of a B cell receptor and antibody is very similar, okay? It's just one has the membrane bound, one doesn't, okay? Alrighty. Now, this is the key thing I wanted to point out. Recall that the T cell receptor cannot see the antigen, while the B cell receptor can easily spot the antigen on its own, and it requires no help. It can just see it, okay? So let's say we are in one of our lymphoid tissues. Let's pick the lymph node, where our resident lymphocytes are, such as our T cells and our B cells. And the B cell happens to come across a specific antigen, okay? Let's say this is an antigen from COVID-19 virus, okay? What ends up happening is, through various mechanisms, all right, this COVID-19 virus, right, the antigen of it will end up on or inside of that lymph node, okay? And the B cell receptor will pick that up. It'll see it. So it'll bind to that B cell receptor. And then this binding will initiate the activation and proliferation, meaning cell division, of this B cell. Okay? And this B cell will produce hundreds and hundreds of clones of itself, meaning additional B cells, that are very, very specific, that have B cell receptors very specific to this antigen and nothing else. Okay? Now, this is where they stop. They don't become plasma cells yet. Okay? The T cell, what about it? Can it see that antigen? No. So this is where our good friend the dendritic cell comes in, which I said has a function called antigen presentation. On its surface, it has this protein molecule called the MHC, which stands for Major Histocompatibility Complex. Okay? There's different types of this, but for this purpose of the video, we don't need to know that. This MHC, okay, will express the antigen that this dendritic cell found. Okay, now is where I answer how this guy found it and why he's doing this. The dendritic cell's job as a resident tissue cell is to sit inside of that tissue, and because it is like very star-shaped and has like large extensions of its plasma membrane, it does something called macropinocytosis, which we talked about in our plasma membrane video. Basically, it does a non-selective drinking of the cell, of the fluid around the cell, okay? And if it happens to catch a microorganism or a pathogen, it'll take that in itself, break it down into its little constituents or antigens, okay? And then it'll take that antigen, put it on this MHC, and express it on its surface. Afterwards, it will start traveling to the lymph node, okay? And it'll go lymph node from lymph node to lymph node, trying to find a T cell that has a T cell receptor specific to this antigen and this MHC, okay? Once it does that, once it finds a T cell that can perfectly see this antigen and this MHC that we are presenting to it, it will then bind to this, it will become activated, and it'll start proliferating. And you will get either a cytotoxic T cell and a helper T cell, okay? But in the process, you'll always make memory cells. And I told you guys why beforehand. Now, once the helper T cell is made, it will then come here and tell those plasma cells that we said, hey, hold on, don't differentiate, those B cells, the hundreds of copies of B cells that we made, that we said, hey, hold on, don't differentiate yet into a plasma cell. The helper T cell will now give the say-so. He'll say, yes, you can do this. You can now turn into a plasma cell that secretes antibodies that are extremely specific to this antigen, okay? And then it'll do that, all right? So you see how specific this response is? They always have to be specific to this, okay? Now, what is an antigen? Again, we've described it many times, but a formal definition. An antigen is a fragment of a pathogen or any foreign substance that initiates an immune response against itself. It's usually a protein, but it can be other things. Now, why do I have a picture of a bridge and the dendritic cell over it? Because the dendritic cell is a bridge between the innate, and let's change colors, is a bridge between the innate immune system and the adaptive immune system. Why do I say this? I say this because the adaptive immune system will not be activated without the innate, aka without the help of the dendritic cell. So watch this flow of thought. Without the dendritic cell, we cannot activate the T cell. And if the T cell is not activated, then cell-mediated portion of the adaptive immunity is completely done. But what about the humoral part? we said that the B cell receptor can just easily see the antigen, which means we can still get antibodies, right? Wrong. We need the dendritic cell to activate the T cell, and then the activated T cell makes the T helper cell. And the T helper cell will go and give the second activation to the B cells to make the plasma cells. So without this dendritic cell, which is the bridge between the innate and the adaptive, we have got no adaptive. Okay? That's why this guy is so important. So important. Okay? All right. To emphasize my point, let's finish it off with this scenario. Let's say that we have a resident tissue dendritic cell in the kidney, okay? And it takes an antigen that it extracted from a pathogen, right? And it moves to the nearest lymph node, okay? It moves to the nearest lymph node by going through a lymphatic vessel. Okay, because remember, every single tissue in your body has a lymphatic vessel that is connected to the interstitial fluid. Okay, and so these guys can easily just migrate into there. Now, watch this. You'll come in here, right, and slowly make its way. Now, something important to mention is that when the dendritic cell is just kind of sitting inside of the tissue doing macropinocytosis, it's called an immature dendritic cell. But as soon as it comes in contact with the pathogen and grabs its antigen, it is now a mature dendritic cell. Okay? And so this dendritic cell, it is slowly moving through this lymphatic vessel towards a lymph node. Okay? And why do I say slowly? Because the flow inside of a lymphatic vessel is extremely slow. And this is, again, what contributes into that very slow activation time of 3 to 5 days for the adaptive immune system. Okay? But anyways, the mature dendritic cell is moving and moving and moving, and it'll eventually make its way into a lymph node. Okay, now mind you, this is just one example, but this guy, he will move from lymph node to lymph node to lymph node to try and find any T cell or any B cell that has, any T cell only that has a specificity to this antigen. Okay, because if the receptor, the T cell receptor, is not specific to this antigen, then nothing will happen. This T cell will not be activated, and this dendritic cell will have to go and find another guy. Let's give an example. There is 1, 2, 3, 4 T cells here currently. Naive T cells, all of them are naive, inside of this lymph node. Okay? This guy, dendritic cell, will go and interact with each and every one of them. He will antigen present to every one of them. Out of these 5, only this one guy has a T cell receptor that is very specific to our antigen. The other ones are either not very specific to it or just completely not specific to it. Okay? This one, though, is extremely specific to it. And so he will bind completely with the MHC and the antigen being presented. Okay? And again, this is the T cell receptor. And he will become activated. Okay? He will become activated. Now, when he becomes activated, right, this naive T cell, he can turn into a CD4 positive or CD8 positive T cell, right? So cytotoxic helper T cell. Okay? And what is happening here? There's a name. Whenever a specific T cell, okay, is selected, right? So whenever the lymphocyte with a receptor that recognizes this specific antigen is going to proliferate, right? Meaning that it will start making hundreds and hundreds and hundreds of copies of itself. This process is called clonal expansion, or another word for it is clonal selection. Okay, so clonal selection or expansion. Okay, same thing. And in this process, you guys obviously know that we'll make some memory T cells. Why would we make memory T cells here? Because if this same pathogen that activated this dendritic cell comes back, right? You know what's going to happen? We have already made memory T cells that are partially activated, and they're very specific, right? They have very specific receptors to go and find that pathogen, okay? Which means the second time we get infected, we will not have to wait 3 to 5 days to activate the immune system if that infection is the same pathogen as the first time. So let me draw a quick graph for you, okay? If this is time, okay, and this is activation, right? The first time we activate, it takes, let's say, 3 and then 5. This is in days, okay? And there you go. It takes about like 5 days to hit the peak, right? But if we get infected, so this is infection 1, okay? But if we get infected with the same X1 again, right? The second time, it looks like this. It's so much faster. In fact, it'll probably be even faster. It'll be like this, okay? The reaction of the adaptive immune system, it'll be so much faster. Why? Because we have those memory T cells, okay? And as we'll talk about, those memory B cells now, okay? Now, same thing that we were talking about here with the kind of specificity, okay? Has to occur here. The antigen will find its way in the lymph node, or it'll find its way into the lymph node, okay? Or the pathogen will find its way to the lymph node, and it'll be phagocytosed, and then kind of killed, and the pathogen will then, the antigen will then be seen. Either way, antigen will find its way here. The antigen will only bind with the B cell that has a specific B cell receptor only to this antigen. That is called specificity, okay? There are, for example, billions of these lymphocytes, and they all recognize different antigens. Only the one with the right B cell receptor will bind to this antigen and undergo activation, proliferation, clonal expansion, and selection, okay? So let's say that it is this lucky guy right here. This B cell will go ahead and bind, okay? And he will go through the clonal expansion and the selection, right? Clonal expansion and selection, okay? And he will start secreting, he will turn into a plasma cell after, of course, the CD4 positive gave the say-so, okay? He will turn into a plasma cell, and he will start secreting antibodies. Now, keep in mind, these antibodies, they have the exact same specificity for that antigen as this original receptor, okay? That's really important to understand, all right? And of course, we will form a memory B cell as well for the exact same reason I gave here, okay? Because next time around when we see this exact same infection, we can respond so much faster, Okay, that is the concept of immunological memory. That's the concept of acquired and adaptive immunity. Okay, that is why you don't get sick with the same exact pathogen twice. Okay, all right. Now, the last concept that we are going to talk about is going to be inflammation, and then we'll finish off the video because we've talked a lot about everything regarding to immunology. So inflammation, as I'll have you remember, is part of the innate immune response. Okay, this what we were talking about here, this was adaptive response, okay, which was initiated by the mature dendritic cell. So inflammation occurs at the tissue. It occurs in the tissue, okay, and it's first initiated by a macrophage. So a resident tissue macrophage initiates the process of inflammation, okay, and it is done when the macrophage is activated. Macrophage activation, okay. When this macrophage becomes activated, it starts releasing things. It releases chemokines and cytokines. Cytokines, chemokines. Okay, what are chemokines and what are cytokines? Cytokines are molecules that can directly kill microbes, so that's one. And two, they have many other functions that I'll list here. And some of them may or may not happen during this inflammation process, depending on the severity. Chemokines, like I told you, they're there to attract other cells to the side of their production. A very common one is interleukin 8, which is used to attract neutrophils. Okay? But nonetheless, this guy will secrete chemokines, all right, which goes and tells these cells, these immune cells in the blood, to kind of come outside of the bloodstream and come into the tissue. And the neutrophils will come out, and the monocytes will come out, and you guys know the monocytes will turn into macrophages when they come into the tissue from the circulation. And by the way, the process of which they are coming out, squeezing themselves out, is called diapedesis. Diapedesis. Now, these guys, once they come over here, they will start to kind of proliferate a little bit, okay? And then they will go and do their functions, which is phagocytosis. Okay. Phagocytosis of what? Of the bacteria, the infection, or whatever it is, okay? Now, these cytokines, like I said, they can directly kill the microbes, right? But depending on their level, they can do other things as well, okay? They can stimulate release of things called acute phase proteins from the liver. And these acute phase proteins include complement proteins. Okay. And by the way, these complement proteins, like I said, they're made by the liver. They're always circulating inside of our bloodstream in an inactivated form, waiting to be activated by some event like this. But when I say that they stimulate the release of complement proteins, they stimulate more, okay, release of even more. And this other thing called C-reactive peptide, okay, which again is just, it can harm bacteria, okay. They can also lower the plasma concentration of iron. You might be thinking, why? Well, because certain bacteria can use iron to divide. That's why. They can increase heat. So you guys know how you'll get a fever sometimes when you are sick, right? That's called, that's not called. It is due to two things. It is due to the fact that these guys, these molecules, these cytokines, they can go to a portion of your brain called the hypothalamus, and they can stimulate heat production there, and they can increase metabolism of your tissues directly, okay, which would increase heat. Now, because these cytokines have the ability to increase heat and causes fever, we call them endogenous pyrogens. Pyrogen. Pyrogen means just the ability to generate heat. Okay? Alrighty. And an example of these cytokines, there are groups of them. Some of them are interferons, some of them are interleukins, some of them are TNFs. TNF stands for tumor necrosis factor. Interleukin, commonly abbreviated as IL. Okay? But they can carry out any of these functions. And finally, the last function is that they can either stimulate clotting or anti-clotting. Okay? And stimulating clotting is kind of good for inflammation because you can wall off the inflamed area and prevent too many things from getting in and out. Okay, so that's what cytokines can do that are released from this macrophage. The second step in inflammation is mast cell activation. Remember, mast cells are the other kind of tissue resident cells, okay, innate immune cells. And when mast cells are activated, they release a molecule called histamine. And histamine can do many things, okay? Histamine can cause vasodilation, okay, meaning the diameter of your blood vessel gets larger, thus more blood flow comes in, okay? It can increase your capillaries' permeability. Now, this can lead to two things. This can lead to, and so can vasodilation, it can lead to the leakage of complement, which is good because that can then activate the complement system. But this guy can also lead to plasma leak, right? Like this, plasma leak. Now, this plasma leak will lead to something called a localized edema. This is what causes this symptom of swelling. Edema due to plasma leak. The redness and the heat are due to the vasodilation. Because remember I said blood is responsible for temperature regulation, and the blood is also red. This is why. Okay. What can the complement leak do? Well, it can lead to complement activation, which we know leads to the membrane attack complex formation, like this, which can lead to cytolysis, okay, and opsonization, which can do what? Increase phagocytosis, right? So here is that complement protein that's opsonizing this bacteria and increasing phagocytosis rate by these phagocytes, okay? So increasing phagocytosis. Beautiful, okay? I should note that most of these here are less at a local tissue level, but they're more so at a systemic level, like whole body system-wise, okay? And again, it just depends on how much of these leak out from the tissue into the bloodstream and start going into the system, okay? If the inflammation is big enough and prolonged enough, then yes, it can definitely happen, okay? But guys, anytime any infection occurs, okay, if it starts getting in the tissue, which it usually does always, what ends up happening is that inflammation always starts, and it's always the same process, but severity can vary, okay? And while all of this is happening, the dendritic cells are sampling, and they are moving their way into the closest lymph node to go to the to go and activate the adaptive immune system, so that this guy, this innate system, with all of its kind of stuff, the complement, the interferons, these cells, they're all working together here, okay? The dendritic cell will go and call for backup, like the big brother, the big guns to come, so that these guys don't have to keep waiting and getting defeated, okay? Because while this is a very fast response, it's not really strong, okay? Because it's not that specific. But these guys, they're really specific, okay? They can help a lot, all right? And the last thing I will tell you is that, of course, the CD4 positive and the CD8s will go to the site of infection to fight the pathogen, right? But the B cells will stay inside of this actual lymph node or wherever they were made, whichever lymphoid tissue they were made in, and they will just keep secreting plasma cells, antibodies to go into the bloodstream and then make their way into the tissue. Okay? Alrighty. Guys, we covered so much with regarding to the immune system in this video. We covered like pretty much an introductory immune system course here, okay? So if you've learned something from this, if you truly learned, if you truly enjoyed it, please hit that like button, subscribe, and leave a comment down below, okay? Thank you so much for sticking around. I will see you in the next video.